I would like to, at this time, introduce Alison Hazel. Alison, our grant recipient, is Greenwood Wildlife Center. Greenwood is a very large wildlife rehabilitation center. I think many of you are probably familiar with it and have delivered some animals there who need care. I know I have. They treat treat thousands of animals every year of about 200 species. And their previous avian clinic supervisor and supporter is Dr. Alison Hazel. She's speaking to us tonight about wildlife rehabilitation, the big picture. So she received her degree in veterinary medicine from Cornell University, which is familiar to all of us. And Dr. Hazel, please take the floor and welcome.
Okay. Before I start, thank you for that. For the 2024 numbers are 4,300 birds and mammals. So those are injured, sick, and orphaned birds and mammals that we treated. Of those 2,023 songbirds, that is a lot of birds. And from the waterfowl, 478. So we're treating a lot of animals. As a veterinarian, I have a little bit of a different view of rehab, perhaps than the public. And so what I'm inspired about for wildlife rehab relates to this concept, the One Health concept, which you're probably all familiar with. It's been around 20, 30 years, and it's very simple. It is the fact that human health is connected to animal health, is connected to ecosystem health. And wildlife rehabilitators have known this for a very long time. Injured or ill or poisoned or sick animals coming into rehab, they can kind of be, a canary in the coal mine for, what is the health of the surrounding environment. So I'm going to tell you a couple of things of the bigger picture of why rehab is important. Okay, an easy one is that we wildlife rehabilitators, we protect the public from disease because people will do crazy things, right? I mean, they will pick up baby raccoons. They will do things like this with bats. They will attempt to raise wildlife themselves, and they can be rabies vectors. They could have mange. And so the fact that we exist and other wildlife rehabilitators exist for a place for wildlife to go is keeping the public from doing crazy things by trying to raise them themselves. Rehabbers or rehab data is a great way to know early about emergent diseases. Most people, I'm sure, are familiar with when West Nile encephalitis hit our shores, 1999. It was a huge wave that went across the country, killing many, hundreds of thousands of corvids. The first way that came to be found was that wildlife rehabilitators were getting called, and literally people were like, dead crow in my backyard, sick crow, and it'd be dead by the time they'd be off the call. And those birds were brought to the attention of DEC and then also the pathologist at the Bronx Zoo. It was that rehab result that rehab data that helped us figure out, oh gosh, we've got a foreign disease. And you're probably aware that the actual disease was figured out by our own Colorado Nick Komar up at CDC. And so that was because rehabbers were calling. House finch conjunctivitis, everybody knows about that disease, but the way it was figured out that it was Mycoplasma gallisepticum was all the rehabbers, including Greenwood. We've sent a lot of finch swabs to different researchers around the country. And then avian influenza, that's a big deal, and I'll talk a little bit about it on the next slide. So we're a great monitoring tool. All these animals that are coming in, and so I have to comment on high-path avian influenza. It's had a huge impact on Greenwood over the last couple of years. It's had a huge impact on me personally, our team, and anybody else who's doing any kind of wild bird or certainly waterfowl work. How we're handling this is that we are testing every adult bird that comes in if it's going to be admitted into Greenwood. And so until we get that test back from CSU showing that they're negative, we have to isolate that bird. And that's tricky tricky. We do not have a large amount of isolation wards. So if we're going to admit the bird, they have to be negative. I will tell you that we're not rehabbing much adult waterfowl because almost all of them are coming in positive for avian influenza. I thought it would end about 2 years ago. We're at 2.5 years now, and birds, it's still, very common and having a big impact on waterfowl. The babies we are rehabbing, ducklings. So if you're under the age of 21 days, those ducklings appear to be resistant, and so we are still rehabbing them. So another thing, wastewater ducks. We send info about ducks that go into wastewater ponds or sometimes petroleum facility ponds to Fish and Wildlife. Those numbers are looked at. And you're probably aware that in the winter, when there's snow on the ground and it's cold, there's no open water for ducks. So what do? In the state of Colorado, they're not required to cover municipal wastewater as in other states, and they land in there. So we'll get these ducks there. They have toxins on them. They have zoonotic diseases. They can sometimes have oil on them. And so that's another illness of wildlife that we monitor. And of course, we do a lot of lead testing, and it's still a problem with waterfowl. We will have neurologic animals come in, though right now, most of those neurologic animals unfortunately have avian influenza. I have to throw this in because I have a captive audience, and this is a slide from March 2025. It is not a slide of bird avian influenza, but this is the mammal slide. I don't know if you guys can see what animals are over there, but the point is the fact that avian influenza has jumped into our mammal wildlife population is what's keeping everybody at CDC, USDA, and wildlife veterinarians like myself up at night. Because we've seen this avian, virus jump to wildlife. Cats are especially susceptible, and we do test mammals at Greenwood if they have signs consistent with avian flu. This is kind of an interesting thing. If you remember, when the cicadas emerged, it was making the news all around the country. Birds were dying on the east. Songbirds were dying. What was that? Well, a rehab center, DuPage Wildlife Center, and the veterinarian there took a lot of those dead birds that came into rehab, and they were looked at for pathologically. And it turned out that why they were dying was they had severe vitamin A deficiency. I mean, who would have thought that eating a bunch of cicadas would give them that? But typically, the diet of a songbird is going to be varied. So they were eating 100% cicada, which has no vitamin A, day after day after day after day, which resulted in vitamin A deficiency. And so their eyes were swollen. They couldn't see that, they ran into things. They had neurologic signs, and it was young growing birds, which of course would make sense, right? That they would be the most susceptible to a vitamin deficiency. So that was some really great work that rehab center did. So if you're a bird and you come to Greenwood, or you bring a bird, there's a 60% chance that you're going to be euthanized. Now that might seem grim to some of you and depressing. It's not to me. But one thing that all of our bodies of animals who have died, they go to the Museum of Nature and Science. So those bodies are not going to waste. Muscle biopsies are taken. Volunteers open them up, look for parasites. Various things are done with them. And why it doesn't really depress me is that if you can pick up a bird, I'm not talking about a baby now, but let's say an injured adult robin, and you can bring it to us at Greenwood, which is fine to do that. It's got a pretty serious problem. And so if humane euthanasia is what 60% of the time we do, it's okay. It's a good thing. It's a treatment, and they're not suffering a slow, painful death. I've been doing rehab long enough to know that things are different, and the data out there, I mean, it really could be mined for support for climate change issues. I mean, we see animals earlier than we never saw before. Squirrels, raccoons, they're breeding earlier. We see species that we haven't seen 20 years ago. We're getting ringtail cats in at Greenwood for rehab. And if you like loons, as you all in this group probably know, that's good because you're going to be seeing them in the winter. And, we used to never see loons. These are the Audubon data maps that show by 2080 how species are shifting. One of the species that's shifting are loons. They're spending the winter here, where we used to never see them again. So the rehab data is very important for looking at changes in specific species in the region and kind of when they're arriving and when they're appearing in Colorado. So what species do we end up getting? I get this question all the time. And the answer is, of course, very easy. It's the species that live near people, of course. Out in the far wilderness, people are not bringing us those animals. And so, yes, it's common species. And I also, and this is not a problem, and it is what we expect to do. We are not rehabbing individual animals to make an impact on the population. It makes neither a negative, it doesn't make a negative impact because the numbers are so small, and it doesn't make a positive impact. The reasons for rehab are these things that I'm going through right now, the kind of the bigger picture ideas. And the reason people are also motivated, besides to help the individual animal, is that if you look at why they come into Greenwood, it's almost always human related. So, of course, cat attack, hit by a car, a tree being cut down. It gets even bigger than that, of course, like habitat destruction. And, ethics towards animals changes and progresses over time. So this idea of let's just let nature take its course, or let's just leave it there, it is really not acceptable anywhere near how it was 20, 50 years ago. And especially the fact that the things that we see in Greenwood are almost completely related to some kind of conflict with humans. So people are motivated. Yes, we do see pigeons, one of the most admirable birds, of course, on the planet, as I'm sure you guys know. And we sometimes people are like, that's stupid. Why are you seeing pigeons? Pigeons are completely unprotected. There's no rules or laws about them. But I do like to mention what's known as the pigeon paradox. And if you've ever read the book Suburbia by John Marsloff, he's an ornithology professor at the University of Washington. Great lecturer, great book to read. There's a chapter in there about this paradox in regards to pigeons. And what it's about is that it was a research around the world that looked at what makes people love birds and specifically develop an environmental ethic that makes them want to protect birds, protect their habitat, save them from going extinct. And interestingly, what they found, because most people end up living in cities, is that the person who has physical contact with a bird, so the person who has the pigeons all over them, roosting on them and feeding them cracked corn, that we might say that person's crazy, that close interaction with a bird and seeing how beautiful they are and looking at them eye to eye actually is the strongest thing that can develop an environmental ethic towards the preservation of birds. So the moral of this little story that I'm telling you is that I think, if the public comes in with a bird, a robin, it's a common species, a house finch, and if Bill Gates walks in with a pigeon, you bet Greenwood Wildlife is going to rehab that animal. And honestly, with the pigeons, we tend to farm them out to new rehabbers. They're great to practice on. They're a very hardy species. So yes, we do birds that are common and maybe not protected and non-native, obviously. And so, let me just quickly look here. Okay, the problem of having and needing rehab is only going to get worse. This is us from space, Cheyenne, Wyoming on the left, then Fort Collins, then Denver, and then we've got Colorado Springs on the right. And so the Greenwood answers at least 10,000, maybe 15,000 wildlife calls a year. What do I do about the woodpecker pounding on my house? What about this? What about this? And so CPW loves us, Animal Control Officer loves us, because we are really trying to educate the public of how to live without conflict and supporting of wildlife. So, we need a lot of rehabbers. We need more. Greenwood can really not keep up, if you'd like to know. And yes, we do the common species, but we really have treated hundreds of species of birds. And so we have to do the common ones. We can't wait around for the special ones to come in. So we're kind of, doing it all. And if your birders, your ornithologists, your animal scientists out there, you'll get asked in your neighborhood, what do I do? I found this, I found that. Please send people to our website. We've got a lot of information about, how do you first figure out, is it a raptor? Because they would, of course, go to Bird of Prey Foundation. And there's a lot of info of what do you do. So it's a great resource to send people there. And we almost never rehab goslings because we reunite hundreds of them. So Canada geese, another very admirable species on our planet. Let's see if this plays, but they are one of the species that will take babies. And so it takes a little bit of work. I don't know if it'll play, but this is some of the stuff that makes it fun, makes it all worthwhile for us. Canada geese, all they want is to get baby goslings away from evil humans. And so they will take them. Do not do this with a mallard duckling, because a hen mallard will drown any duckling that is not her own. And then, this is what makes it all worthwhile for us, inspires us. And like I said, if you like loons, they'll be coming in the winter. Nice. So thank you again for your support, and I might have a couple minutes for questions. But thanks again to DFO for supporting us. We greatly appreciate it.
Thank you, Allison. We do have a question. Fran asks, or says, foxes were not on the list of mammals suffering from avian flu. Are they immune?
They're not immune. It's a great question. But we've tested some neurologic foxes, so I don't know the specific numbers. There's not currently right now, we're not finding any quote unquote immune mammals. Now we've got it down to mice that are positive. So foxes, it seems like the canine group, cats are getting it much more than dogs, but dogs and canines, foxes, coyotes are getting it less, but are not immune. Sorry about my noise here.
Very good.
Any other questions?
Yes, we have one more question. Do you rely on federal funding?
Excellent question. We get no federal funding. We get no state funding. We rely mainly on donations and support. We do have some municipal contracts where we will take orphans, let's say baby raccoons, and from certain municipalities in Boulder County. So they kind of contract us so that their teams don't have to deal with it. But we are mainly public supported, and Amanda Stout, who's also listening, she's our grant writer. So we rely on grants, but not federal funding. So thank you for asking, because that we'd be in a pickle if we were relying on federal funding.
Excellent. I think that's the last question we have for you, and we
Sure.
want to thank you, Allison, and you, Amanda, for both being here this evening. And we will move on to our next speaker. I'm
Thank you.
pleased to introduce DFO grant recipient Holden Fox. Holden is a PhD student at CSU in Fort Collins. His talk is Conservation Genomics of Loggerhead Shrikes, and we all love shrikes. This is a partially migratory grassland species that has experienced alarming population declines. However, there's not enough information about its migratory patterns. So Holden has been gathering and analyzing genetic information from Loggerhead Shrikes from 5 Western states, and he will discuss his research progress to date. Holden, take it away.
Thanks. Thanks for having me. All right, let me see if my presentation will get up here. I'll go into present mode. Why not? Can everyone see my presentation okay? Yeah.
Yes.
Was that a thumbs up? Okay, great. Super. Okay, so my name is Holden, and today I'm presenting on conservation genomics of Loggerhead Shrike. So I grew up in a town called Everett in Northwest Washington. So it's about 30 minutes north of Seattle. And then I got interested in birds in high school. So I spent a summer sailing in the Salus Sea there around Washington and British Columbia on this sailboat called the Orion, pictured here. And during that summer, I saw lots of seabirds. And I just thought they were incredible, and they sparked an interest in birds pretty early on for me. And so I went on to study biology at Lewis and Clark College in Portland, Oregon. And so there's Lewis and Clark there in the left. And there I think while I was studying biology, and I just became interested in biological computing specifically. So like the computational aspects of working with big data in biology. And so Lewis and Clark has a computing cluster, which is shown in the center. It's called BLT because there's 3 compute nodes. There's bacon, lettuce, and tomato. And there I got started using the cluster to make phylogenetic trees of spider families, actually, which is pretty neat. Okay, and so after undergrad, I spent a year working at the University of Arizona in Tucson, where I studied brown and Arizona brown and brown recluse spider venom under Matt Cortez and Greta Binford. And Tucson also had a bunch of great birding. And so here's a brown recluse spider in the top right. I have one of a 3D structure of one of the molecules, these venom proteins that we are working with. And then this on the bottom is a big nuclear magnetic resonance spectrometer, which is an analytical chemistry tool we use to measure the rates of activity of these molecules. And so after that experience, I came back to birds, and I started a PhD at Colorado State with Kristen Bruig. And here I use genomic tools to improve conservation outcomes for loggerhead shrikes. So there's Kristen on the left, and then on the right is me holding the first bird we caught in our western sampling trip last May. And so a little bit about shrikes. They're grassland birds, and they're found in grassland and shrubland and then pasture habitat all across North America. And as I think Susan alluded to, they're partially migratory. And so in the northern part of the range, they're obligate migrants, which means that all individuals in a population they're migrating every year. And then at intermediate latitudes, the birds are partially migratory. And so in a population, you might have one bird that remains resident year round while another individual, it migrates. And then at southern latitudes, the birds are year-round residents. And this range also spans a gradient and seasonality. So at the northern latitudes, the environmental conditions they fluctuate more widely than at southern latitudes, right? And loggerhead shrikes they're of conservation concern. They face drastic population declines across the range. And so since breeding bird survey estimates are available, they've declined 76% range wide. And so this is 3/4 out of all individuals since we have data for. And the population trend's been negative across the board as you could see in these population trends on the right. But the intensity of the declines, they vary across the range. And this is evidenced by um, these northern migratory populations. They've suffered some of the earliest declines. And so shrikes, they used to occupy this area in gray in the upper Midwest, Northeast region, but they've largely been extirpated in recent years. So mostly just a small population on Ontario remaining for that region. And because of widespread population declines, the species has been listed by the IUCN as near threatened. There's two subspecies that are federally endangered. So there's the subspecies in San Clemente Island in the Channel Islands in California. And then there's also a subspecies in eastern Canada, those Ontario birds that are federally endangered. And then they're also state endangered or threatened in 14 US states. They're not actually threatened in Colorado, although those population trends are still negative here. And so for this chapter of my PhD dissertation, I'm really interested in how adaptive units are distributed across the shrike breeding range. So we'll talk a little bit of genomics, hopefully not too in the weeds for everybody. All right, let's do it. So conservation geneticists, we generally quantify variation in the genetic code of populations using markers in the genome, these genetic markers. So I use a genetic marker called a SNP, which is just a point mutation where the identity of a single base pair, so an A, a C, a T, or a G, they differ between individuals in a population. So in the example on the left, there's 3 birds, and then at this position where the SNP is, the purple bird has an A, the blue bird has a G, and the orange bird has a T. And so I do this across every variable position in the genome. So bird genomes they're around 1 billion base pairs long, and my data set has around 1.5 million SNPs, so 1.5 single base pair mutations to work with. And then this section on defining adaptive units is focused on adaptive genetic variation. So I'll briefly break down like adaptive and neutral mutations. So yeah, basically we can split all these positions in the genome into either adaptive or neutral loci. And the adaptive ones are just those that they affect traits that impact fitness. So they're under natural selection, and the neutral ones are not. So adaptive variation is important for species survival. We know that small populations face a heightened risk of extinction due to the extinction vortex. So this process leads to reduced genetic diversity overall and a loss of evolutionary potential, which hinders populations' ability to respond to disturbances like climate change. And adaptive genetic diversity, so variation in all those those loci that affect fitness, that's what determines a population's evolutionary potential. And so preserving this unique adaptive genetic variation across populations is important for survival of threatened and endangered species overall. So how do we manage populations to preserve adaptive genetic variation? Adaptive conservation units are one way to guide conservation decision-making. And we do this by identifying populations that are adapted to similar environments. So in this figure, we have several individuals, which are these white dots, that make up two genetically distinct groups in blue. And they're distributed across, this is an environmental gradient. But we can subdivide them into four distinct adaptive units based on, so all the variation in the genome that's associated with environmental conditions. That's associated with their local environment or their fitness. And so delineating AUs, it helps us preserve adaptive genetic variation specifically. And I'll briefly go through this, but generally partially migratory species in the northern hemisphere, we see this pattern of populations at higher latitudes, they're more likely to migrate, and the populations at lower latitudes are more likely to be resident. And this is the same pattern we see at Shrike, right? The northern populations are the migrant ones, and the southern populations are the residents. And then for migratory species more generally, whether a bird migrates or remains resident is thought to be an adaptive trait. And so some previous work was done on burrowing owls, and they measured the strength of correlations between environmental variables and the genome, and they found that yeah, these birds they differentiate based on whether the birds are migrant or resident. And so no need to really diagnose this figure here. I'll just keep chugging along. And so my question, this part of my work is how are adaptive units distributed, right? The hypothesis is that adaptive genomic variation in loggerhead shrike populations is driven by this gradient of seasonality along that latitudinal gradient. And so my prediction is one that AUs, these adaptive units, they'll correspond with variation in seasonality. So breaks in the genome that are associated with environment, they'll roughly follow this latitudinal gradient seasonality. And then also that the adaptive units, they'll correspond to differences in whether the populations migrate or are partially migratory or are resident. And so if that's the adaptive trait, whether they migrate or not, that the AU designation, they'll correspond with the migratory phenotype per population. Okay, so the first step really for me was whole genome sequencing of loggerhead shrikes across their range. And so the range is pretty big. It's a difficult task. And so myself and many collaborators, we collected about 436 right now breeding genetic samples. And so on this map, each point is a sampling location. And then this includes sampling in those areas where we have endangered populations. So the purple arrows point to samples from the endangered Ontario population and also samples collected. These are museum samples actually collected from the San Clemente Island population in the Channel Islands. And so DFO was really instrumental in allowing me to do this range-wide sampling. And so these purple circles that are popping up on the screen, they show the places that I went this so 1 year ago now to collect birds. And you can see that the sampling is pretty dense there because, yeah, the rest of the range, we get partners that send us feathers, they send us blood samples for where they're doing monitoring. But when we go out on our own to do targeted sampling, you get really, really good sample sizes in those areas where you want. And so for each of these birds, I sequenced whole genome DNA at an average of 5x coverage. So whether it's blood or it's a feather and there's skin in the skin cells in the tip of that feather, we sequence DNA and then sequence or we extract the DNA and we send it off to a sequencing facility to do the genomic sequencing. Okay. And so this is my main result right now that we see the population structure or the differences in the genetics of these populations across the range. They correspond to migratory phenotype. So this is a PCA or principal component analysis of total genomic variation. So this is these neutral loci and the adaptive ones that are associated with fitness. And the PCA, generally, each point represents the genome of an individual bird. And the genomes that are more similar to each other, they cluster more tightly together on the PCA. And so they're just more related to each other. And so these breakpoints that we see in the clusters, they're the beginnings of conservation units, really. And so the results, they suggest that shrike populations, they cluster by migratory phenotype, whether the bird's a full migrant on the left, a partial migrant in the middle, or these two resident populations in the Gulf. These orange points are the Gulf Coast, and these purple points are more like Florida and the Southeast birds. Yeah, they cluster with migratory phenotype. So suggesting that my hypothesis is supported. And then currently I'm working on defining the adaptive units in Loggerhead Shrike. So these are the just the regions of the genome that are associated with fitness and with the environment. And the way this gets done is with a genotype environment analysis, which is a really cool study. I'm working on this right now, so I'm using Canada warbler as an example data to illustrate my point. But we use climate and land cover information, vegetation information, and we put them through genotype environment association testing to find out which regions of the genome are associated with the environmental predictors. And so you get an output like this where individual Canada warbler genomes, they separate based on their associations with different environmental variables. And so the green populations, they're more adapted to variation in precipitation, and then the blue populations, they're more closely associated with tree cover, and then these south populations, and if that's like pink or red, those are more associated with temperature and precipitation variables. And so it's these kinds of relationships that I'll use to delineate the adaptive units in Shrike, allowing us to preserve different subpopulations that have unique adaptations to their environment that will be helpful in preserving evolutionary potential in these birds and overall improving survival outcomes in Shrike. So thanks to everybody who's contributed to the work. Special thanks to collaborators up in Canada, Amy Chabot, the Loggerhead Shrike Working Group, both Moore Lab of Zoology's Natural History Museum and the Natural History Museum at San Diego, and then DFO, among other funding partners who've been instrumental. And I'll leave it open to questions.
Thank you, Holden. Very, very interesting. We're glad you're doing this research and happy to fund it. I don't see any questions in the Q&A right now. I'll just remind the audience, if you have a question for Holden or any of our speakers, please post your question in the Q&A feature on your Zoom desktop. Okay, and seeing no questions, I think we will move on. Our next speaker is grant recipient William Churchill Anderson, a PhD student at the Department of Ecology and Evolutionary Biology with CU Boulder. He's going to talk to us tonight about the continued exploration of a novel avian hybrid zone. Will is studying the 2 subspecies of house wrens that occur in Colorado and their hybrid zone. Understanding hybrids contributes valuable information, of course, about speciation and genetic divergence. So, Will, take it away.
Great, thank you. Can you all see well? Cool. All right, so thank you guys for coming. My name's William Anderson. I'm a second year PhD student at CU Boulder in Scott Taylor's lab. And I'm going to be talking about the second chapter of my dissertation on the genetic basis of divergence in a North American songbird, which is the house wren. So I am by trade a evolutionary biologist. So I seek to understand speciation because that is one of the most fundamental goals of research in evolutionary biology. And so many of you are probably familiar with speciation, if not by name. So speciation is the process that gave us all of the Hawaiian honey creepers. So originally, millennia ago, a small group of rose finches from Asia were blown over to the Hawaiian Islands, and then through the process of speciation, they diverged to become all of these different species of honey creeper. And this pattern is repeated throughout the natural world. We also see it in African cichlids. So these Rift Valley cichlids originated as these single or few species that eventually radiated out to become these really speciose groups. And so understanding the underlying mechanism of this process is something that I'm keenly interested in. And so one of the most common processes for speciation is something called allopatric speciation. So this occurs when you have an original population, so for example, rose finches that get blown over to Hawaii, and they're on the main island. And then at some point, some event occurs to divide that population. So we can say that a storm blows half of the rose finches to Kauai. When they're geographically isolated like that, processes like natural selection and genetic drift and gene flow within these populations, but not between, results in the generation of things called reproductive barriers, which basically are any trait in the bird that or in any animal that could end up impacting reproduction between populations. And eventually you'll get your own specific species, these separate groups. And this is commonly mediated by glaciers in North America. So Pleistocene glacial expansion and retreat is a common mechanism of allopatric speciation, especially in birds. And a classic example in North America are the pocile chickadees. So we have all of these chickadees in North America. They were all pushed into these different refugia by these glaciers. So as the glaciers moved down and kind of covered all of Canada, most of the habitat was then pushed further south as well. So most of North America was Arctic tundra, and only small regions were protected and had suitable habitat for all these birds. So the ancestor of all these chickadees was fragmented into these glacial refugia, and in that refugia, they became these different species. And many of you guys are familiar with the bottom two center birds here, the black-capped chickadee and the mountain chickadee. And they provide a really interesting opportunity for studying speciation. They're not the subject of my talk, but they are the reason that I'm here, because that's mostly what our lab studies, is the hybridization between these two birds. And that is really where you can start getting at speciation. So hybrid zones are natural laboratories for studying speciation. So when you have these birds, so now, for example, these bullocks and Baltimore orioles, when you have these birds that are pushed into these separate geographically isolated refugia by glaciers, once you remove the glaciers, we currently don't have glaciers. We're not living in an Arctic tundra right now. Those glaciers have retreated, and as they retreat, habitat and subsequently the birds therein expand back out. And eventually they'll come into contact with each other, and that's called secondary contact. And we live very close to the range of secondary contact between these two birds, which is like Tamarack Ranch. I'm sure you all are familiar with the tons of hybrid birds there. And so in this secondary contact, we can test the barriers that are developed in the glacial refugia. So between Baltimore and Bullock's orioles, the Baltimore oriole developed this all black head, and Bullock's oriole developed this white wing patch. So they have these visual barriers to reproduction. So they can identify each other separately and say, that is not of my species. But again, as we see at Tamarack, these barriers are not entirely perfect, right? There's a lot of hybridization occurring. And this hybridization is actually really common. These are 3 examples of east-west avian hybrid zones that we find on the Great Plains here. Each of these have developed differing levels of reproductive isolation. In the center, we have black-headed and rose-breasted grosbeaks, and those birds really rarely hybridize. They were all developed, all of these different species pairs were developed in the same glacial refugia, but they just diverge to different degrees. And so we have these really rarely hybridizing rose and black-headed grosbeaks. And then on the left, we have passerina buntings, which I'm sure all of you are familiar with the extensive hybridization that occurs there. But again, they're still considered to be species. And then on the right, we have the flickers. And those are not considered to be species currently, but again, you can see that in these glacial refugia, they developed these differing color patterns. And that's an important thing here, is the reproductive barriers in all of these birds that we've mentioned previously have been visually apparent. You can tell what a species is based off solely its plumage. In a lot of cases, these birds also exhibit differences in call or song as well. But recently, work actually out of Kristen Rueg's lab, as well as out of Alberta, has found that some cryptic populations, so birds that look really similar to each other and sound really similar, are actually diverging as well. So on the left, we have Swainson's thrushes, on the right, we have warbling vireos. And so there's actually significant genetic divergence in these populations, but there's no difference in plumage. And so what is driving that? That's kind of the basis of all my questions. What drives this difference? In these populations, it's things like migratory divides. So on the right, these vireos, they both are migratory. Both subspecies of warbling vireos migrate. But the western warbling vireo returns to the breeding grounds about 2 weeks earlier than the eastern warbling vireo. So the western warbling vireo has already begun reproducing. It has been setting up territories, building nests, finding mates for 2 weeks by the time the eastern one gets back. So they kind of have this temporal isolation from each other, and they don't really interbreed. Hybridization is pretty rare. And so my whole work has been on house wrens and answering similar questions in house wrens. So what is driving divergence in house wrens? As you can see here, these are 2 subspecies of house wren, Troglodytes aedon parkni on the left and Troglodytes aedon on the right. You can tell that they're both house wrens, but you can't really tell that they're either one of the subspecies. We've done some work that I won't present here today showing that it's actually impossible to tell them apart by plumage. There are no differences in plumage between these groups of birds. And yet they exhibit pretty significant differentiation in their genome. So that's approximately 4% differentiated in the mitochondrial genome, which is about 2 million years. So for 2 million years, they've been kind of an independent evolutionary lineage to some degree. But house wrens are really widespread. All of you should be familiar with them. They probably nest in your yard. They'll kick the chickadees out of your nest boxes. And the species that we have in the US and Canada is the northern house wren now. They were recently split up. So the ones south of the Isthmus of Tehuantepec in Mexico are Troglodytes musculus now. But in the north, I focus on 2 of the subspecies, which is Troglodytes aedon parkmanii and Troglodytes aedon, one in the east and one in the west. And there are no vocal or morphological differences. So a bit of background on these birds. Not a ton of genomic work has been done on them. So during my undergrad, I identified extensive hybridization between both Troglodytes adon parkmani and Troglodytes adon. And this is the sampling map. So I collected a bunch of birds from Colorado, as well as the West Coast and the East Coast, and I compared all of their genomes. And so you'll see here, I'll show a PCA, which was nicely presented before me. And so in the top left corner, in that blue cluster, those are all birds from the West Coast. These are Troglodytes adon parkmanii. And then the top right, there's a cluster of red birds, and those are all Troglodytes adon from the east. So there are two separate genetic clusters. And all the birds in the middle with a gray fill and then multicolored rings around them, those are birds from Colorado. And so you can see that they fall intermediately. So this is genomic data showing that these birds are intermediate between the east and the west. We also generated something called hybrid indices, showing the genomic background of the ancestry of each bird from Colorado. So on the far left with a value of 0, a hybrid index of 0, are the Western Troglodytes adon parkmanii. And on the right in red, again, is Troglodytes adon, that eastern bird, with a hybrid index of 1. And so the way that this works is if you take a Troglodytes adon parkmanii and a Troglodytes adon, so that eastern versus western bird, and you create an offspring from that, it would have a hybrid index of 0.5, so a fully intermediate genotype. And as you can see, most of our birds from Colorado here have this fully intermediate genotype per this data, which is pretty interesting considering this looks like an example of isolation by distance. You have one bird in the west, and it just gradually becomes a different bird in the east. Except birds from Nebraska are more closely associated with birds from New York than they are to Colorado. So there's something going on with genetic divergence here, where we have these 3 different groups. And so what I wanted to do is explore the differences between these 3 groups and understand what underlies the divergence here. So they're morphologically cryptic, they look the same, and they sound the same. So why are they still different? And so to do this, we did whole genome sequencing, calling SNPs again, like explained before. We have these polymorphic sites in the genome that show differences. And so we're just comparing differences here. And so we took a new data set with 5 birds from the west and 5 birds from the east, representing that western population and that eastern population, and then 20 birds from Colorado, where it seems likely that there is this intermediate population or a hybrid zone or something going on. And what we did is we, again, generated this PCA. And you can see that our birds from Colorado cluster really nicely in the top left corner in gray, and the birds from the Pacific cluster in that bottom left corner in blue, and the birds from the east cluster in the top right corner in red. However, a couple of the birds from Colorado actually cluster with the Pacific individuals or are intermediate, and that's going to be important soon. We also generated estimates of something called FST, which is a fixation index, and I'll explain what that is here. It measures how different populations are, and it also varies from 0 to 1. So if you have a group of birds that all have A at one site and a group of birds that all have T at that site, they would have an FST of 1. They're fully different. All of them are different from each other. If they all have A, FST at that site would be 0. And if half of them have A and half of them have T versus the other half has A and the other half has T, then the FST would be 0.5. So it's just a measure of how different these populations are. And the reason it's called a Manhattan plot is because you plot it by the location on the genome. And so you'll have mostly low levels of genomic divergence, and then you'll have these little spikes of differentiation where, let's say, there's something really evolutionarily important, like migratory behavior or something like that. You'll have these big spikes of differentiation, and it looks kind of like the Manhattan skyline, hence the name. So what we did is generated one of these plots, and as you can see, we have a very poor man's version of Manhattan. Most of our buildings are very low-lying. We don't have a lot of differentiation across most of the genome. We have small peaks in every chromosome here, which is the number on the y-axis is the chromosome. Those peaks most likely represent something called a centromere, which is the middle of the chromosome, and that does not generally recombine, so it becomes pretty different, but it's not necessarily evolutionarily meaningful. However, we do kind of have this giant region of elevated FST, so a really different region on the Z chromosome, which is the avian sex chromosome. And so if we zoom in there, we find a really sharp break where we have really low-lying FST, so no differences in most of the Z chromosome, and then this big jump up, a big plateau of highly differentiated genome. And this is indicative of something called a chromosomal inversion. So if we think back to high school biology, we have recombination between chromosomes. And sometimes what happens is the chromosome is just fully flipped, and that's called an inversion. So during that recombination, you have one that reads one direction and then becomes rotated and so will be read in the opposite direction. And that will mess a lot of the time with recombination and reproduction in species. But it's also a really helpful evolutionary tool in some sense, where any genes within that are then locked. So not a lot of selection can really happen in those regions because they're inverted. So a lot of the times really important genes to selection and to evolution are within those inversions. So for the orioles, if you think back to the orioles with all those color plumage patterns, there are genes related to color vision within the chromosomal inversion in orioles. So they're very, very helpful and very important a lot of the times for differentiating populations. And so what's also kind of novel about this is that more than 95% of the differences between the eastern and western house wrens is found in the Z chromosome. So more than 95%. So in the instance where all birds have an A and all birds have a T, where they have a FST of 1, more than 95% of the FST values of 1 are on the Z chromosome. So most of the chromosome is not differentiated, or most of the genome is not very differentiated, whereas the Z chromosome is highly differentiated. And so what we can do is something called local PCAs. So we run that same PCA, that principal component analysis, on specific regions of the genome, and we can observe patterns. And what we see is on the left for that main first region of elevated FST is the same pattern as before. So almost all of our power in the PCA, so all of the differentiation between these birds, is coming from this region. And what we have interpreted from this is that there are actually 3 kinds of house wrens in North America, not just 2. You have your western bird and you have your eastern bird here in blue and here in red. And then you also have a Colorado type. There's an interior west bird that is genetically different from the eastern house wrens and from the western house wrens. And you can see that there are birds that are intermediate between the gray cluster, that Colorado group, and that blue cluster, the western group. Those are birds that are heterozygous for these haplotypes. So they have one type from let's say from their mother that comes from Colorado, and one type from their father that comes from the West Coast. So they're not fully segregated in their ranges, but there are 3 different types. And this is actually a really common biogeographic pattern. We see it, as you guys are all probably familiar with, we see it in blue-headed and plumbous and Cassin's vireos, right? So in the east, we have the blue-headed vireo. In the interior west, we have plumbous vireo. And in the west, we have Cassin's vireo. This also holds true for sapsuckers. So we have yellow-bellied and red-naped and red-breasted sapsuckers following the same distributional patterns, as well as white-breasted nuthatches. So we have an eastern form of white-breasted nuthatch, an interior west, and then a west coast form. So there are actually 3 species or 3 populations of house wrens in North America rather than just the original 2 as thought. But now, getting back to the original kind of question, now that we know there are 3, what are the differences there between all of these birds? We want to know why they're different and what is driving this differentiation. And we find 2 pretty fascinating potential avenues of exploration here. The first being relating to elevation. So we basically created a list of all the genes that were in that inverted region, that really high differentiated region. And within that, we found it was highly overrepresented for metabolic genes and genes relating to high elevation. So it's possible that the birds in Colorado, because they're living at these high elevations, have this different inversion type to keep them safe and operating at maximum efficiency in the mountains, which is not necessary at these low elevations. It's also possible that it has to do with migratory behavior, because there's a gene called VPS13A in that inversion, and that's the only gene that has been found to influence migratory direction in these vermivora warblers, so golden-winged and blue-winged warblers. So right now, what we're doing is expanding our data set because we only have 30 birds. So I've actually caught birds all the way from Grand Junction to Kearney, Nebraska. And now we're going to really expand on this to really elucidate which one of these 2 processes is responsible for differentiating these birds. So in summary, the western and eastern subspecies of house wren show significant genomic differentiation. There are 3 distinct genotypes of house wren in the US and Canada. So our birds here are different from the ones on the East Coast and from the ones on the West Coast. And the majority of the differences between these birds are located on the Z chromosome. So I would just like to acknowledge and thank everyone who helped me, all the funding sources, and the great scientists who have helped me put this all together. And then I will take questions.
Great job, Will. Thank you so much.
Of course.
I am in the Q&A, and I am not seeing any questions right now. So, I'm just going to give it a second here. Oh, okay, here's a question from Nathan. How do you differentiate the subspecies if there are no plumage or vocal differences?
Fantastic question. If you're a taxonomist from the 1800s, you just potentially write some stuff down about colors. The way that they were originally described was one had a rufescent wash and the other didn't, which doesn't hold up on specimens, or we did spectrometry on the birds. None of it really holds up. There were some claims about differences in measurements, so leg length or bill length, and those don't hold up either. So realistically, you wouldn't tell them apart. They all should be considered one species, but with these three distinct genetic populations. So probably there should just be one subspecies, one species of house wren rather than these multiple groups. Yeah, it's kind of a case of oversplitting.
Okay, thank you. And we have one more question. Where did you get your t-shirt?
Oh, nice. I'm not sure. I'll tell Susan, you can maybe pass it on.
Okay. Thank you, Will, very much. All right, moving on. Let's see. Our next speaker and our big finish for the night is Emily Breaker. Emily is the collection manager of vertebrate zoology at the CU Museum of Natural History. And if you're familiar with the museum, they have a very large ornithology collection, and that is what Emily is here to talk about tonight. So Emily, welcome, and you have the floor.
Well, thank you so much. Let me share my screen. How's that looking? Oops, sorry. One moment. Good. All right. Well, thank you so much for having me. Happy to talk about the ornithology collection here at the CU Museum of Natural History. And we actually had our first DFO field trip to the collection last year, and we're planning a repeat trip in June. So we hope that some of you can join us for that. So I wanted to start off by saying that behind every exhibit lies a collection. So, in addition to putting specimens on display for the public to enjoy, most natural history museums are active centers of research that has substantial specimen collections behind the scenes. So when you go to a museum, generally you're really only seeing a very small proportion of our holdings. So the CU Museum of Natural History was established in 1902. And in addition to the vertebrate zoology collections that I manage, we also house paleontology, entomology, invertebrate zoology, botany, and anthropology collections. And our footprint extends over 3 different buildings on campus. But here in the vertebrate collections, we have approximately 130,000 specimens from across the 4 vertebrate disciplines listed here. So we house reptiles, amphibians, fish, mammals, and of course, birds. But our herpetology collection is definitely our largest collection and most actively utilized resource. So our ornithology collection has about 15,000 cataloged specimens, and the vast majority of these are preserved as study skins. So you see those on the slide. So this is really the traditional unit of comparison used for research, but we do also house skeletons, preserved eggs and nests, frozen collections, and taxidermy. And the collection dates back to 1839. So our oldest specimen is featured here. This is a bufflehead duck, which I think looks great for being over 180 years old. So our collections do predate our establishment, and that's simply because the museum made a lot of purchases at the time that it was established. So in terms of geographic scope, our bird collection contains species from around the world, and these holdings really just reflect different curator interests over time or different expeditions. So for instance, we do have a series of about 22 species of hummingbird from Colombia. We have over 160 specimens from Thailand, and we have over 50 taxidermy mounts purchased in Japan in 1904. But the vast majority of our collection does hail from US localities, and over half of our collections are from Colorado. So we have really strong regional representation of birds that occur across the state's diverse elevation gradients and habitats. In terms of taxonomic breadth, we have 931 species present in the collection, with the majority of our holdings in passerines. So that's pretty reflective of bird diversity more generally. And we have particularly strong representation in sparrows, especially juncos, New World warblers, rosy finches, and just great coverage of North American families in general. But we do have a bit of everything. So we do have kiwis, we have penguins, we have albatross, and we have a very large stuffed cassowary. So very many well-known charismatic taxa from around the world. So in addition to our historic holdings, we are actively growing the collection and adding new specimens regularly. So we do that in a couple different ways. One is through targeted fieldwork, where we're going out, we're asking a specific research question where we sacrificially collect a limited number of birds. We also partner with agencies like CPW, Fish and Wildlife, Boulder, OSNP. We work with wildlife rehabilitators. So we've worked with Greenwood in the past, we've worked with birds of prey. And so as was mentioned, sometimes they have to euthanize injured wildlife, so those come back to the museum. We do occasionally get donations like historic taxidermy, and we also do partner with outside researchers that are bringing in field collections. And then finally, we salvage. So we have statewide permits for the state of Colorado where essentially we're allowed to pick up dead wildlife. So that could be roadkill, that could be window strikes, cat kills, and roughly 90 to 95% of our current growth results from this sort of passive salvaging. So instead of something dying and decomposing, it can sort of live on in the collection and be used for research and education into the future. So I wanted to kind of zoom out and really talk about the role of biological collections more broadly. And so one of the primary roles of collections is that they serve as archives and repositories for Earth's biodiversity. So, you might be wondering, why do we have so many specimens? And one of the answers is we're documenting and confirming a species' presence by archiving it as a voucher specimen in the museum. So sometimes people might think, do you really need another robin or another grackle? And we say yes. Each individual really does represent a different data point, and certainly in a time of climate change, habitat loss, zoonotic disease, you really can't anticipate what's going to happen in the future. What's common now might not be so decades down the road. And we know that from artifacts that are coming out of ancient Egypt and from taxidermy that's been preserved since the 1500s in Europe, we can assume that if we do maintain our collections in archival quality conditions, that they will survive on the scale of centuries, if not millennia, into the future. So what can we tell from our specimens? So when we remove an animal from the wild, we record the date and the specific point on the Earth's surface of where it was collected. And that individual really serves as a time capsule or a snapshot of its range, what it was eating, its phenotypic expression, its reproductive status, its age, its color morph, the diseases and parasites it was carrying, all of which is really reflective of that individual's specific genetic makeup, but as well as the spatial temporal context from which it was collected. So even though we have 200 northern flickers in the collection, each one of those is going to be genetically distinct, just like you and me, and collected at different points at different times. So this is a schematic that really shows the constellation of information that we can gain from each specimen, both the physical samples as well as the types of digital data that we can associate with individuals like sound and video recordings. So another role of biological collections is basic and applied research. So collections have really long been the foundation of our understanding of life on Earth. And really provide the raw material to explore questions of ecology and evolution and study patterns of biodiversity across space and time. So our museum's main claim to fame really is facilitating what's known as retrospective research. So by taking, a large specimen series collected over the course of decades or centuries, researchers can really examine animals from the past and compare those to animals from today and really quantify, how a particular variable has changed over time. And so in that way, we can really use collections as historic baselines, and we can investigate a multitude of things, but really common themes are, examining range and trophic shifts, the patterns of biological invasions and disease, how species are responding to climate change, and even things like environmental toxins. And birds in particular are a great system since most species tend to molt once a year. So their feathers are a perfect sample from that year that they were collected. So researchers can look at contaminants like heavy metals, microplastics, soot concentrations, and use specimens essentially as biomonitors for pollutants in the environment. Another role of biological collections is reference material. So specimens are the basis for virtually all scientific illustration. So figures within academic papers, to Audubon's famous works, to field guides that we know and love. So artists, of course, are going out into nature, observing animals and their behaviors, but the thing about living animals is that they move, they're skittish, and so those same artists are absolutely coming to collections to look at specimens up close and really get an eye for textures and fine details and close-ups of their anatomy. So next I wanted to share some historic highlights from the collection. So the first is our egg collection, and that represents our most historic holdings, with the bulk of it assembled between 1850 and 1900. And that was really during what's known as the Victorian egg craze. So at the time, a lot of museums were sponsoring hobbyists to go out and collect egg clutches. Also, wealthy people were too, to have some egg clutches to display in their cabinets of curiosity. And these specimens are typically accompanied by really rich data that often indicates, how many eggs were in the clutch, what species of tree a nest was found in, the nest height, the materials it was composed of. And that really gives us a lot of insights into ecology and also phenology. So when the birds were reproducing 120, 170 years ago, and how that timing is really shifting with warming temperatures and coinciding with peak resource availability as our climate changes. So in general, birds tend to be laying earlier in the year. Another historic highlight are specimens collected and prepared by Charles Aiken, primarily in the 1800s. So he wasn't academically trained, but he did a lot of survey work and documented birds throughout the state. And he's really considered to be one of Colorado's pioneering ornithologists. And he collected about 4,700 specimens in his lifetime and prepared them as skins and taxidermy. And he ran a small taxidermy museum out of Colorado Springs. And so his collection was purchased by the Colorado College in 1907, but that museum eventually folded in the 60s, and that collection came to CU. And so it's a really historic and important early collection of Colorado avifauna. We also have rarities in the collection. So we certainly have endangered species, but we also have extinct taxa, including species like Eskimo curlew, Carolina parakeet, and passenger pigeons. And so this really goes back to our role of archiving life on Earth. And I'm sure you all know that the passenger pigeon was once our most common species in the US, and they were said to blanket the sky by the billions. But along with habitat destruction, these guys were overhunted to their extinction, essentially by the turn of the 20th century. We also have type specimens, and a type specimen is the first species or subspecies of its kind named to science. So it has a corresponding type description that's published, and it recognizes the new species and the Latin name assigned. So we often refer to types as the name-bearing specimen. And so we always say that in an emergency, should it be safe in a fire or flood situation, we would save the types. And that's really true across museums. So our cabinets are on wheels. We have 7 types in the ornithology collection, but we have 300 types in our herpetology collection. So these are really important collections, scientifically speaking. Next, I was going to talk about specimen preparation. So we do this once a week with our students. So our most common prep type is the study skin, and unlike posed taxidermy, these are really compact forms that fit nicely in a drawer, and they're easily compared. So when we prepare a study skin, we make an incision down the belly. We then remove all the soft tissue, organs, and internal anatomy. We then stuff the skin with cotton and usually insert a dowel rod to sort of reinforce the specimen. Then we pin it out to dry, and it dries for about a few days to a few weeks. But study skins really capture the outward appearance of an individual, and it preserves things like plumage, including molt and color and external anatomical features. And so, historically, morphology and plumage were exclusively used in alpha taxonomy to define species limits and understand sort of the natural variation that occurs within a species. But now we combine, of course, examining these external physical traits with molecular technologies to look at DNA and really delimit species and understand where they sit in relationship with one another on that phylogenetic tree. Spread wings are another preparation type that we make. So with the study skins, their wings are folded, so you can't really examine them. So in this way, we remove all the muscle tissue and then pin the wing out in an extended position. And this really allows us to look at patterning and assess aging by looking at molt limits. And we can also calculate different aspect ratios to assess things like wing loading, which gives us insights into things like flight behavior, effort, and dispersal ability of different species. We also prepare birds as skeletons and fluid specimens, so you'll have to forgive my picture of a mammal in this talk, but this is all I have on my phone. This is a skull of a bull moose in our beetle colony. So once we remove a carcass from the skin, we use beetles to actually feed on the muscles and produce a clean skeleton. So we soak the skeleton at that point in different chemical baths to degrease the bones, and then we number each one individually with a catalog number so that we can always associate each bone back to the individual it belongs with, and of course the correct collecting data. And then fluid preserved specimens are injected with formaldehyde, and then we fix the specimen that way, and that really prevents the proteins from breaking down post-mortem. Then we transfer the specimen to ethanol for long-term storage, and ethanol is just a less toxic chemical, and it acts as a preservative, so it prevents bacteria and mold from growing in the jar. And what's great about fluid preservation is that you have the complete specimen, so it retains all of its internal anatomy. So if someone down the road wants to dissect that specimen and examine, a specific organ, look for parasites, pathogens, they can do so 50, 100 years after the fact. For every specimen, we take tissue samples, so we tend to pull heart, muscle, and liver so that they can be made available for genetic analysis. We also keep parasites that we encounter, so things like ticks, fleas, lice, parasitic worms. And symbionts are interesting because they really can impact both individual and population level health. We also examine stomach and crop contents since they represent important ecological associations, and we might even keep those samples, especially with raptors, when we're able to positively ID prey items. So a lot of times rodents or lizards or birds can be identified from their skeletal elements, and we'll catalog those separately in the collection. So specimen data is really just as important as the physical voucher. So unfortunately, there's really little research value to a specimen that lacks data, especially if we don't know where or when it was collected. So this is a sampling of the data points that we collect for each bird as we prepare it. We take several external and internal measurements, and we also assess things like reproductive condition, molt, fat deposition. So you might be familiar with some of these if you've done banding before. And we record all this information in tiny handwriting on a tag and also in our database, which really allows us to bring together all the information known about each specimen and any data products generated from that individual through time. So these are just kind of two partial record examples that I have. So on the left, this is a northern bobwhite record. So you can see it's collecting data up at the top. The specimen has been cited in a publication, so there's a link out to that. It also has been sequenced, so those DNA sequences are linked in a different database called GenBank. On the right is a record for a Heerman's gull This guy had chewing lice, so we saved those parasites, and so those guys are linked. We've got photos of the wings. We have all the trait data, locality information, and the samples, so researchers can know, oh, there's tissues available and request those. And then all of this data is publicly searchable online in our database Arctos, so there's a link there. And we actually also push this data out to a couple of global databases that aggregate and compile data across the world's museums, which really enables research at a global scale or big data. And so this way, a researcher can search across all museums simultaneously in order to borrow specimens, or even analyze all the collated occurrence data ever recorded about a given species to really understand its range and distributions through time. In terms of accessing and utilizing collections, researchers can schedule a visit to examine specimens in-house for a specific project here on campus. We also loan specimens out to different museums so people can request loans and examine our specimens more locally. And we fulfill dozens of loans each year. We also provide course loans to several CU classes, so ornithology, which Will TAs, archaeology, all sorts of the ologies for across our vertebrate disciplines. And so that really gets students inspired about birds and other animals by really being able to handle them and observe them up close. And I just wanted to show a few examples of the hundreds of publications specifically linked to bird specimens here at CU, and give you an idea really of the different entry points to specimen-based research. So of course, taxonomy and systematics are a mainstay of the collection and why collections were originally developed. But there are many, many other avenues of research possible beyond these traditional applications. And we really have a broad user base. So some people aren't even ornithologists, right? They might be parasitologists or epidemiologists. And so that's a really a great function of collections, is that a specimen collected for one study can be used again and again for totally different kinds of research. And so many of our specimens, for example, were collected a half century before the discovery of DNA. And these same specimens can be included in molecular studies. So as new technologies emerge, new research trends emerge, we can really go back to that raw data and ask new questions of it. So it's really exciting to think about, what advances will be made 20 or 50 years from now, and really thinking about the utility of our specimens through time. And so I just want to end on some ways to get involved if you're interested. So as I mentioned, we do hold permits for the state of Colorado. So if you find a dead bird in good condition, you'd like to donate, just make sure to freeze it immediately and then just email me, and you can drop that off at our front desk anytime during business hours, which does include the weekends. But we do need your name and contact, where you found it and when. We also host observation data in our database. So we will accept photos, video, and sound recordings, but we're not trying to replicate eBirds, so we don't really have the bandwidth to do that. So it's really kind of limited to things that are unusual or a bit out of the ordinary. So range extensions, hybridism, other interesting variations. So this is a leucistic black-capped chickadee from one of our faculty curators from her bird feeder. And then this is actually an example of nest competition that we observed on a DFO field trip, where the nest appeared to have been constructed by a robin, and then grackles came in and laid some eggs and they were squabbling. And so we got that picture. And then finally, we have some volunteer opportunities. So feel free to email me if you'd like to come and help us with some curatorial tasks. And we also have field note transcription. So we're at the age where a lot of our students can't read cursive. So we need help with transcribing some of these sort of travel logs from our early collectors. So yeah, please email and be in touch if you are interested in any of these opportunities, and I'll take time for questions.
Emily, thank you. It never occurred to me that cursive would be a bit of a snag for people in your profession, but I can see why they would be. That is really interesting information. Thank you so much for sharing that. We had a couple of questions that you already answered, so I won't repeat those, but terrific presentation, and we really appreciate it. I don't see any questions in the Q&A. Let me look in the chat just quickly. Nope, I think Jim had a question, but you answered it already when you talked about how you treat the specimens when they come in. So that answered that. I want to thank you, Emily, and keep up the good work, and thank all of our speakers tonight. It's been a really educational experience, and we appreciate all the work that you all do. I also want to say to all of our guests tonight, all of our viewers, thank you for coming, and please, please get yourself up on Dinosaur Ridge. Think about volunteering for DFO, and please think about making, if you're so inclined, please think about making a donation to the Hawkwatch team or our grants program. And I want to thank Jill Boyce, who leads the grants program at DFO, and her grant program volunteers. She has several volunteers who help her read grants and decide where the money goes from our grants program. So thank you to all of our speakers this evening. We really appreciate your presence here, and we hope we will see you again sometime. And I think we're ready to wrap it up. Oh, I want to, one thing, there was a question earlier from a participant asking about Greenwood's most unique species that they have treated. And before she had to leave, Alison responded, it is swans. So there's the answer for that question.